Hello. On this small animation, we will address what are extracellular vesicles, also known as EVs. We have a donor cell in blue and a recipient cell in red that will communicate through these EVs. EVs form within the lumen of a multivesicular body, but large EVs can form within the plasma membrane. The multivesicular body can also fuse with the plasma membrane and release these small EVs. If we take one of these small EVs to check into its contents, we can see that they have as cargo lipids, protein, and nucleic acids. And this material will be received by the recipient cell through different mechanisms, including membrane fusion, ligand binding, and endocytosis mechanisms as well. Thank you. Hello, I would like to present the work Impact of Oral Anti-Diabetic Drugs on Gut-Derived Extracellular Vesicles, Proteomic Signature. If you have any question that is not addressed during this flash talk, you are free to contact the corresponding authors. Metabolic homeostasis results from the interaction among all organs, highlighting inter-organ communication for a healthy energy balance. For the case of type 2 diabetes development, gut dysbiosis is a key contributor and extracellular vesicles affects disease progression. We have previously unveiled that gut-derived EVs isolated from a high-fat diet model, when administered to a healthy animal, drive a pre-diabetes phenotype, resulting in weight gain, glucose intolerance, and hepatic lipid accumulation. Every year, there are about 3.7 million deaths due to diabetes and high blood glucose. For this reason, in our lab, we have the long-term aim of unveiling the role of EVs in diabetes and pre-diabetes. On this occasion, we are focusing on anti-diabetic drugs metformin and pioglitazone, and we have the objective of assessing the impact of oral anti-diabetic drugs on gut-derived EVs proteomic cargo. Methods. Mice were kept on normal chow diet, that is a healthy diet, or high-fat diet that will induce pre-diabetes. Metformin and pioglitazone were orally administered by gavage to the diabetic mice for 6 weeks every day. Mice were then sacrificed and guts were extracted to isolate extracellular vesicles. After LCMS, proteomics analysis was performed. Liver tissue samples were also analyzed. After 6 weeks of treatment with the oral anti-diabetic drugs, control mice presented lower body weight compared to all the other groups, and mice treated with metformin had a reduced body weight compared to those treated with pioglitazone, and the same is seen in the clinical settings. Importantly, both drugs improved glucose intolerance. A principal component analysis was performed on the proteomics dataset, and the results suggested that the treatment samples were similar as of those of healthy mice and quite different from the high-fat diet ones. The volcano plots of both high-fat diet versus treatment with metformin and high-fat diet versus treatment with pioglitazone allows visualization of top-regulated proteins. The Venn diagram shows 50 proteins shared among upregulated pioglitazone and upregulated metformin proteins. On this heat map, we can see a column comprising 3 samples corresponding to each group. Seeing the results from the liver histology, both metformin and pioglitazone improved liver steatosis compared to pre-diabetic animals. The heat map shows that proteins associated to lipid transporter activity were shared among normal chow diet and both treated groups. On the contrary, proteins related to acyltransferase activity, which plays a fundamental role in the decision of whether fatty acids are stored, were upregulated on high-fat diet. We unveiled that treatment with metformin or pioglitazone resulted in a protein signature that resembles normal chow diet, that is the healthy signature, and the top-regulated proteins for treatment with metformin and pioglitazone are related to lipid metabolism. The take-home message will be that both these drugs can modify the protein content of gut-derived EVs and ultimately play a role on inter-organ crosstalk for metabolic homeostasis. Thank you.